Welcome, ladies and gentlemen, to another in our series of the Professor Speaks lectures. I am very pleased today to introduce Professor Paula Tennant. It's taken a little over 2 years to get Professor Tennant to agree to this, so we're really pleased to have her here today to talk about at least one of the things she loves, and that's about viruses. It's a topic that's apparently very near and dear to her, and she does a lot in her research and in her teaching on the topic and in the study of viruses. Professor Tennant earned her BSc in botany from the University of the West Indies, Mona, here in 1990. She went on to obtain her PhD in molecular plant pathology from Cornell University in 1996. Following this, she held postdoctoral positions at Cornell University for the period 1996 to 1997, and the Biotechnology Center here at the University of the West Indies at Mona for the period 1997 to 2000, and then joined the faculty in the Department of Life Sciences in 2000 as a lecturer. She later became a senior lecturer in 2008 and a professor in 2012. Professor Tennant's research focuses on identifying viruses and the viruses that affect vegetables and fruit crops. Her work emphasizes the development of management strategies using biotechnology and exploring natural sources of tolerance and resistance from diverse germplasm, including domestic and wild species. She has authored over 100 publications on virus diseases in fruit and vegetable crops. Professor Tennant also co-teaches undergraduate courses in molecular biology, virology, and plant biotechnology. It is my great pleasure to introduce to you, ladies and gentlemen, Professor Paula Tennant.
Thank you, Dr. Busby Earle. Today we explore the realm of viruses. Our world is teeming with a vast and diverse array of viruses. The oceans alone harbor more viruses than there are stars in the observable universe. Mammals carry some 320,000 different types of viruses. When you consider viruses that infect non-mammalian animals, your plants, terrestrial bacteria, and every other potential host, the total becomes staggering. Many of us perceive viruses as agents of disease, especially given the recent COVID-19 pandemic, but their role extends far beyond this common perception. In this lecture, we will explore the multifaceted nature of viruses, essentially their tale of duality. While they can be detrimental, viruses also actively contribute to maintaining the health of organisms, spanning from plants to insects to humans. Additionally, they are contributors to scientific knowledge in the fields of molecular biology, medicine, and agriculture. They play a vital role in supporting ecosystems, and so this afternoon we delve into this duality. Let's begin with the basics of viruses. What are viruses? For a time, a virus was defined mainly by its size. An entity smaller than bacteria. And so this drawing provides a visual representation illustrating the relative sizes of the viruses. So here are the viruses over on this side. This is your bacterial cell, and this is a human red blood cell. The viruses are essentially 0.1 micrometers across, and so this makes them about 1/10 of the size of an average bacterial cell and a human blood cell. They're about 1,000 times smaller than the width of the human hair, about 5,000 times smaller than a grain of sand. And such a tiny size means that you cannot even spot these viruses using the light microscope. The largest virus, the MonaMedia virus, It's huge. It approaches the size of a small bacterial cell, and it was only described in 2003. We'll say a little bit more about this virus in a bit. Now, the first evidence that viruses exist came from experiments set up to determine the cause of a disease that was plaguing tobacco. In 1879, Meyer, the director of the Agricultural Experiment Station in Holland, initiated work on a disease of tobacco. In the Netherlands and in certain parts of Germany, he reported that the harm done by the disease is often very great and that it has caused the cultivation of tobacco to be given up entirely. Meyer used sap extracted from a diseased tobacco plants as inoculum to inoculate healthy tobacco plants, and he was successful in reproducing the symptoms of the original disease. He subsequently tried to identify the causative agent using techniques and knowledge available for studying bacteria at that time. And so the samples were examined microscopically. Samples were cultured on medium formulated for bacterial growth, and none of the tests were successful in identifying or isolating the causative agent. Nonetheless, Meyer concluded that the agent was a bacterium that had probably lost its activity in the lab. Two other scientists, a botanist and a soil microbiologist, conducted similar investigations into this disease of tobacco. So that's Unveiling in 1890, a Russian botanist. He essentially conducted the same sorts of experiments that Meyer conducted. However, he used porcelain filters. So these filters were available at the time. They were used to purify water. They retain bacteria. He collected the filtrate, rubbed it onto healthy tobacco plants, and Unveiling was able to repeat the original disease. He reproduced the original disease. He reported as well that it was impossible to grow the agent in culture, and his conclusion that it was a bacterium, but an unculturable bacterium. It was the soil microbiologist Bergenic who proposed a revolutionary idea. He posited that the filterable agent was a unique type of pathogen, a new type of pathogen that exists in a non-cellular state. Further, it wasn't until, at the end of the 20th century, the concept of a virus was established in negative terms. Viruses cannot be seen, they cannot be cultured, they cannot be retained by bacteria-proof filters, they cannot be cultured on artificial media like bacteria. And it wasn't until the mid-1930s and the early 1940s, that the molecular structure of viruses was developed. It was by experiments from a chemist, Wendell Stanley, in 1935, that laid the groundwork for the development of the structure of the viruses. So he discovered that the virus tobacco mosaic virus, the same virus that was studied earlier, where it could be crystallized, and in this form it resembled a pure protein with a regular structure. When he dissolved this protein and introduced it into the tobacco plants, it seemingly came back to life, and he was able to induce the disease symptoms in tobacco plants. And so this discovery challenged the distinction between living organisms studied by biologists and non-living molecules studied by chemists. In 1936, Baldwin and co-workers, biochemists, they revealed that the virus TMV actually consists not only of protein but 6% RNA, 6% ribonucleic acid, making viruses more complex than mere chemicals. The virus was later visualized, TMV was later visualized by X-ray diffraction and also photographed by electron microscopy, all in the early 1940s. So what exactly are viruses? The viruses are made up of genetic material, RNA or DNA. This genome is inside a protein shell, often referred to as a capsid. Some viruses, in addition to this protein shell, have an external lipid membrane, a fatty envelope, a fatty layer known as the envelope. And so what we're seeing here is the tobacco mosaic virus. This virus exhibits a helical structure, a very simple helical structure, helical arrangement. We're looking at the protein coat. It's encapsulating the RNA within. This is a rabies virus. In addition to the protein coat, it has the fatty layer. And here we have spherical, quite a number of viruses have this structure, a spherical conformation like your coronaviruses. And what we're looking at here is the lipid envelope surrounding the protein coat. The electron microscopy images shown here illustrate the structures of various viruses. We have our TMV showing the helical symmetry we just described. This virus here it has been magnified 150,000 times. Our rhinoviruses, those viruses that cause colds, the common colds, are spherical viruses. Flu viruses also more or less spherical, but they have a lipid layer. And then there are those viruses that infect bacteria, the bacteriophages, with a much more complex structure, where you see the helical structure of the tail and the spherical structure of the head, sort of a tadpole conformation. But where do these viruses come from? Where do viruses come from? Every year, virology students ask this question. The study of the origin of viruses is challenging, in part because viruses do not leave behind fossils in the traditional sense. They lack cellular structure and they lack the hard parts in many organisms that contribute to fossilization. So scientists have to rely on comparative genomics to study the evolutionary history of viruses. This involves examining the genetic material and looking for similarities and differences between different viruses to trace their relationships. There are 3 hypotheses on the origins of viruses, and it's important to note that our understanding is still evolving. And so there's the ancient origin, escape origin, and reduction origin. Scientists believe that viruses may have been around for billions of years, possibly since the early days of life on Earth. And they might have co-evolved with cellular organisms. So they evolved in parallel. That's the ancient origin. According to the escape hypothesis, viruses may have originated from genetic elements that escaped from cellular organisms, and over time, these elements evolved into the viruses that we know of today. The reduction origin, on the other hand, suggests that viruses were once free living organisms that developed a relationship with cellular organisms and then became dependent on the host. Over time, they lost certain functions as they adapted to a parasitic lifestyle. Each of these hypotheses has merits, but in 2003, new evidence tipped towards the reduction origin. There was a discovery of the giant virus mentioned earlier, the virus. It's a huge virus. It's the biggest. It's about the size of a small bacterial cell. This virus was found in amoebas, which are single-celled organisms. And inside these amoebas were mysterious blobs. They were big enough to be seen through the light microscope. They actually looked like bacterial cells. But when scientists tried to detect bacterial genes in these blobs, they found none. Instead, they detected virus genes. And in addition to these genes, they found genes that would normally be found in cells. They named the blob, this structure, virus. And is derived from mimicking to emphasize the initial misclassification of this virus as a bacterium rather than a virus. But let's go back to this finding of genes that would normally be found in cells. Why would these genes be in a virus? The logical interference is that the virus has them as holdovers because its lineage originated by reduction from a cell. The existence of the virus challenges our understanding of viruses and cells because it blurs the traditional boundary between viruses and cellular life. This large and complex virus possesses features traditionally associated with cellular life. Viruses might not fit our definition of life, but at the same time, they're too reminiscent of life to be described as inanimate matter. Now that we have established what viruses are, let's delve into the duality. And we're going to start first with the dark side of viruses. Viruses have etched an indelible mark on human history, triggering epidemics and pandemics and testing our resilience. Among these are the flu or influenza viruses, and these are often thought of as tornadoes, a sudden disaster that arrives with little warning and does its worst in a relatively short time. 3 of these calamities occurred in the 20th century. The impact varied. There was a devastating 1918 Spanish flu, after which influenza viruses continued to circulate globally. The Spanish flu occurred during a time when medical and scientific understanding of infectious diseases was limited. Communication was constrained, and there were no antiviral medications or vaccines at the time. The devastating impact of the Spanish flu underscored the importance of global cooperation, timely information sharing, and the need for research and development of medical interventions. And then there was a moderate 1957 Asian pandemic, H2N2, and the mild 1986 Hong Kong virus. Another major pandemic was caused by the human immunodeficiency virus HIV. It actually started in the early 20th century but remained undetected until the early 1980s. While effective treatments are now available, the global impact of HIV/AIDS has been profound. The initial response of HIV/AIDS was marked by stigma, discrimination, and a slow recognition of the virus. There were challenges in understanding the modes of transmission, and developing effective treatments. The HIV/AIDS pandemic prompted a re-evaluation of public health strategies. Examples of these adaptations include prevention and education programs which involve the distribution of condoms and increased emphasis on promoting their use to prevent the sexual transmission of the virus. Additionally, the needle exchange programs were designed to reduce the risk of HIV transmission among injecting drug users. Another crucial initiative was the widespread availability of antiretroviral drugs and efforts aimed at making antiretroviral therapy more accessible and affordable. This significantly contributed to improving the quality and length of life of individuals living with HIV/AIDS. The COVID-19, the SARS-CoV, the severe acute respiratory syndrome coronavirus 2 was first identified in Wuhan, China in the late 2019 and spread globally, leading to millions of infections and deaths. The pandemic demonstrated the interconnectedness of our globalized world, presenting challenges in terms of rapid transmission, overwhelmed healthcare systems, and vaccine distribution. COVID-19 underscored the importance of early detection, transparent information sharing, and collaborative international efforts. The development of vaccines in record time demonstrated notable progress in the fields of science and technology. Viruses can also cause harm in domestic animals, leading to diseases and similar levels of economic losses. Prominent viral threats worldwide include the foot and mouth disease virus, avian flu viruses, and African swine fever. Foot and mouth disease is a highly contagious viral disease affecting cloven-hoofed animals such as pigs, cattle, and sheep. The origin of foot and mouth disease is not known. An African origin is, however, suspected. The disease occurs on many African and Asian countries. Its impact can be separated into two components: direct losses due to reduced production and indirect losses caused by costs of control. Outbreaks have led to trade restrictions, as have the avian flu viruses, the H5N1 and H7N9. Outbreaks of these viruses in the late 1990s to present lead to culling or slaughter of animals, trade restrictions, and economic losses in affected regions. African swine fever is another example of how viruses can disrupt ecosystems, economies, and agricultural systems. The virus affects domestic and wild pigs. It can result in a rapid death of almost all infected animals. Outbreaks since the 1920s have resulted in significant losses with culling and trade restrictions in affected areas. The virus is endemic to Africa, and the ability to remain viable for long periods in raw or insufficiently cooked pork products has enabled it, through travel and trade, to spread widely throughout Africa and to other continents. In the second half of the 20th century, there were outbreaks in many African countries, a number of European countries, in Cuba, Dominican Republic, and Haiti, and Brazil, and most recently in 2018 in China. China is the home to half of the world's pig production. It rapidly spread through China, the Philippines, Indonesia, and Papua New Guinea. Corals are also affected by viruses. One coral disease of particular ecological importance is the white plague that infects multiple species of Caribbean corals. The white plague begins as lesions or spots at the base or periphery of the coral and progresses progresses rapidly over the surface of the colony. Giant viruses, giant DNA viruses, the ones that we mentioned earlier, these have been isolated from bleached coral, and they might play a role in the white plague disease in the Caribbean. Black band disease is another disease of coral that is actually the most studied coral disease worldwide. So the disease is characterized by a dark mat that forms a band on the surface of the coral colonies. This mat actually consists of 3 bacterial species: cyanobacteria, sulfate-reducing bacteria, and sulfide-oxidizing bacteria. And recently, viruses have been found infecting members of the cyanobacteria community. The role of the viruses in the disease is not yet certain. Plants are not exempt to virus infections. The FAO estimates that 40% of the global crop production is lost to pests every year. Among plant pathogens, which include fungi, bacteria, and viruses, the major group causing reduction in yield and quality is viruses. Viruses infections can lead to almost 100% crop loss in tropical and subtropical regions. The viruses that infect economically important crops are listed here in this slide. Banana bunchy top is the most impactful, affecting bananas, and there's a citrus tristeza virus causing disease in citrus. We at the UWI have conducted investigations into various diseases affecting our agricultural crops. At Mona, we characterize the citrus tristeza virus infecting citrus in Jamaica, and we noted an increasing spread of virulent strains. At Mona and Cave Hill, they have identified a number of viruses that infect sweet potato, and similar work in Trinidad with tomato and pepper, pumpkin and yams. They've identified viruses affecting those hosts as well. But how do viruses slide? The first step involves the invasion of host cells. They invade their cells pretty much by latching onto the outside of the cell, a bit like a key clicking in a lock. The better the fit, the more likely the virus is able to access the interior of the cell. Once inside of the cell, the viruses seize control of the cell's machinery. They effectively direct the cell to produce more viruses instead of carrying out their usual functions. They manipulate the cell's normal functions to replicate and generate copies of themselves to generate more viruses. And then the final step involves the departure of the viruses from these newly assembled viruses from the cell. The viruses go on to infect other cells, perpetuating the infection cycle, and then they eventually leave the host in search of new hosts. Departure from a host occurs through various means, including the respiratory droplets expelled during coughing or sneezing, releasing bodily fluids like blood or saliva, and in some cases through the fecal-oral contact or skin-to-skin contact. Transmission on the mouth parts of insects have led to that have fed on infected hosts. So how do they actually cause So once they have gotten into the cells and reproduced in the cells, replicated in the cells, the damage that is caused falls under these 3 categories. We can have direct cellular damage, altered cellular functions, and also immune response activation. So the diseases they cause is because of direct cellular damage, altered cellular functions, or the immune response, the activation of your immune response. Influenza virus, for example. These viruses, they primarily infect respiratory epithelial cells. Virus replication damages the cells, and this leads to the respiratory symptoms that we are all familiar with. The infection triggers the immune response of the host in an attempt to stop the infection, and this manifests as symptoms of fever, runny nose, and body aches. But by 6 to 7 days, the body has typically succeeded in clearing the virus from the body, marking the resolution of the infection. The virus finds new hosts by hitching a ride in respiratory droplets expelled from the mouth during activities such as coughing or sneezing. Similarly, with your foot and mouth disease, the animals suffer from blisters or ulcers in the mouth, and this is because of virus infection and damage to epithelial cells in the mouth, the feet, and udder. Cattle generally recover within 8 to 15 days after infection. Infection with viruses of the herpes family. These viruses are responsible for oral lesions of night fever and genital lesions of genital herpes and red itchy rashes that often starts on the face, chest, back in chickenpox. Yes, chickenpox is a type of herpes virus. The infection process of herpes is a bit different. These viruses, they also attack and multiply in epithelial cells during the start of infection, and the infection appears to resolve quickly. However, the virus has actually moved out of this location, moved out of these cells, damaged these cells, moved out and go into the nervous system, affecting the cells of the nervous system. They develop lifelong infection with their hosts. So unlike the flu viruses and the foot and mouth disease virus, these viruses enter into a latent state following the initial infection, allowing them to evade the host immune system and persist permanently in the host. They hide under the radar of the host immune system with almost no multiplication, no replication, and no generation of new viruses. On occasion, there is a reactivation and the virus starts multiplying and returns to the epithelial cells. Reactivation and resulting disease is important for viral spread to new hosts or for the maintenance of the viral reservoir in the host that results in recurrent infections. And so this is regarded as the ultimate virus infection or ultimate relationship between a virus and its host. Disease symptoms caused by plants range from discoloration of leaves, as shown here. You can have yellowing, you can have browning, you can have patterns of irregular coloring of the leaves, as well as the petals of flowers. And here we see a mixture of light and dark green areas or a mosaic pattern. You can also have symptoms of distortion of the leaves, twisting or curling. In addition, viruses can affect the size and the shape and the quality of the fruit or vegetable product. Products from infected plants display visible symptoms such as the lines and ring patterns as seen here on the papaya. These affect the appearance and organoleptic properties of the produce. The symptoms, again, are a result of damaged cells, damaged tissues, and in this instance, in the plants, the result of the impact of the viruses on the chloroplast, the site of photosynthesis. And it can be as well because of the damage to the plant's vascular system, which is responsible for the movement of water, nutrients, and signaling molecules, hormones. This disruption upsets the plant's normal growth and development. A clear example of this is the induced dwarfism of leaves and stems, as well as little or no fruit production caused by the papaya ring spot virus, as shown here in the slide, but also of banana bunch top virus in bananas. Once plants are infected, little can be done to free them from virus infections. But what happens with your bacterial cells, your single cellular organisms? Viruses can infect bacteria, and they induce either a brief infection or a persistent infection, a more prolonged persistent infection. In the brief infection, the virus, or otherwise known as bacteriophages, the phages infect their genetic material into the bacterial cells and takes control of the cells, directing the synthesis of new viruses, and then they leave the cell by breaking open the cell, disrupting the cell, destroying the cell, and they go off to find more bacterial host cells. In case of the prolonged and persistent infection, once the virus has injected its genetic material in the cell, this integrates into the genome of the bacteria, becomes part of the bacterial DNA. The viral genes are then passed on. They're replicated along with the bacterial DNA during cell division. And so persisting and passing on to the progeny for several generations without causing any immediate harm to the cell. However, under certain conditions, the virus can transition to the lytic cycle, that's over here, resulting in the production of new viruses and the subsequent degradation or lysis, breaking open of the host cell. But within this dark narrative, there is a bright side. It now appears that viruses with bad intent, or viruses that cause disease, represent only a fraction of the massive viral community. Technologies of genomics are rapidly uncovering new viruses from seemingly healthy hosts. Take us, for example. It is estimated that 380 trillion viruses are living on and inside our bodies. That is 10 times the number of bacteria. Yes, some cause illness, but many simply coexist in urine or in the blood, for example. Now, seeing that they do not target our cells, what then do they target? They target bacteria. These viruses, or phages, inject their genetic material into the bacterial cell. They use the host cell's machinery to make copies of themselves and then burst out to infect more bacteria, killing their hosts in the process, the lytic cycle. As these viruses move around the body looking for bacteria, some of them bind to cells on the surface of mucosal membranes, such as those that line the nose, the throat, stomach, and intestines. And the viruses do not replicate there, but they lie in wait for vulnerable bacterial hosts to pass by. Studies suggest that these viruses help regulate bacterial populations in the gut by selectively targeting and infecting specific bacteria. They may contribute to the balance of the microorganisms in our body and prevent the overgrowth of harmful bacteria. One phage getting a lot of attention is the Croas phage. Discovered in 2014 in the Netherlands, Studies since have shown that the crossphage inhabits most people around the world. It has not been linked to any disease, and it's believed that they control the presence of the common gut bacterium called Bacteroides. When Bacteroides organisms escape the gut, they can cause significant pathology, oral infections, abscesses in the neck, in the lungs. But once they are in the gut, they play an important role in the digestion of complex carbohydrates. Not only do viruses inhabit our bodies, but they also have deposited their genetic material in our genomes, in our DNA. About 8% of our genome, of our DNA, is retroviral DNA. DNA from retroviruses, and retroviruses are a family of viruses that have a unique feature. They actually possess an RNA genome, but once they enter in a cell, they convert to DNA and integrate their DNA into the host DNA, into our DNA. This integration allows the retrovirus to establish a persistent and long-lasting infection within the host. The viral genetic material becomes a permanent part of the host DNA. About 8% of our DNA comes from viruses that infected our ancestors long ago. Viral code often manifests as inert pieces of DNA, silent pieces of DNA, but in one case, it conferred a new and useful essential function. Evidence indicates that we owe our ability to have live births to DNA that was co-opted from ancient retroviruses that infected our ancestors millions of years ago. A gene that originally helped a virus fuse with host cells and enter into host cells found its way into ancient animal genomes. It was then repurposed in these animals to generate a similar protein that helps fuse cells to create a special structure around what became the placenta. This opened up a possibility in some animals, that of internal pregnancy. It became possible for a female to carry her developing young inside her body rather than leaving them vulnerable in one place as eggs in a nest. And over time, the design of this new mode of reproduction improved and the placenta involved. Paradoxically, some viruses can also confer protection against certain diseases. Case in point, the GBVC virus. Virus C, is a common blood-borne human virus that was discovered in the mid-1990s. It is related to the hepatitis C virus, and it is a distant relative of the West Nile virus and dengue fever virus. GB virus C infection has not been associated with any disease. However, studies have found an association between persistent GB virus C infection and improved survival in HIV-positive individuals and delayed progression to AIDS. Scientists have also found that having GB virus C seems to lower the risk of death in people infected with Ebola virus. Similar protection has been reported with herpes viruses in mice. Herpes virus infection makes mice less susceptible to certain bacterial infections, including bubonic plague and listeria. Infecting people with herpes virus, bubonic plague, and listeria to replicate this mouse experiment would be unethical. But scientists who reported these findings suspect that their findings in the mice likely apply to humans. The protective phenomenon has also been reported in plants. In bitter melon plants, the bacterial pathogen Erwinia induces wilt disease and triggers the production of volatiles in the plant that attract the beetle vector to the plant, facilitating movement of the bacterium to healthy plants. However, when these plants are infected with a virus, zucchini yellow dwarf virus, the production of the beetle attracting volatiles is reduced. And so infected plants infected with the virus do not actively attract beetles that carry the virus, the bacterial pathogen. Plants infected with viruses are also protected against infections from related viruses. The phenomenon is known as cross-protection. Cross-protection was first observed about a century ago by McKinney. He found that infection with a tobacco plant with a mild yellow strain of tobacco mosaic virus prevented its subsequent infection by a severe mosaic strain of the virus. Since then, various research groups have extended this finding to practical applications. There are reports of protection conferred by strains of potato virus Y in tobacco. Mild strains of citrus tristes of virus protect against the severe forms of the disease in citrus species, in sweet orange and grapefruit. Papaya ring spot virus in papaya. Recently, in 2015, mild strains of Pinot mosaic virus were commercialized in 14 European countries for the protection of tomato crops from a range of Pinot mosaic virus strains. Cross-protection is somewhat similar to immunization in animal hosts, but the underlying mechanisms in plants differ because antibodies are not produced in plants. Instead, the mechanism involves the destruction of any virus resembling that of the virus that is used as the protective strain. So in other words, the protective virus triggers a natural defense mechanism in the plants known as virus-induced silencing, and this results in the degradation, the attack and degradation of the incoming virus. So it's pretty much the scenario that's illustrated in B and in C. Based on this discovery of using a virus to induce the host plant's defense, scientists turned to using only a gene of the virus, only a part of the virus, to activate the defense in plants. So in 1980s, Powell Abel and his colleagues demonstrated that transforming tobacco plants to express the coat protein gene, the gene that codes for that outer coat of the virus, tobacco mosaic virus, conferred resistance to the virus. And this led to the development of transgenic or genetically modified plants. Since then, considerable efforts have been invested in developing virus resistance to different viruses affecting crops relevant to agriculture and food production in developing countries. We at the UWI-MONA tried the technique with our papaya varieties and the papaya ring spot virus. However, this project and most other projects elsewhere in developing countries have remained at the proof of concept stage, and few have passed the regulatory system to the stage of approval for cultivation. In fact, only 5 transgenic crops engineered for virus resistance have been approved for cultivation and are commercialized. The reasons that these and other virus-resistant transgenic crops have not made it to release in developing countries include a lack of regulatory framework and strong opposition from lobby groups. But that is a lecture for another day. These challenges posed by regulatory hurdles towards genetically modified virus resistance spurred the development of an alternative technique known as spray-induced gene silencing. Spray-induced gene silencing. Spray-induced gene silencing is based on the mechanisms at work in cross-protected plants and in transgenic virus-resistant plants. Remember, the presence of the virus or protein of the virus triggers the destruction of the second related virus. And so the key, what they have used here is they have used the key to triggering this resistance in plants. That is the generation of double-stranded RNA molecules. And so the spray-induced gene silencing approach involves designing these molecules, designing these double-stranded RNA molecules to trigger the plant's defense. The molecules are complementary to a specific target region of the virus for which resistance is being developed. Once these molecules, these double-stranded RNA molecules, are applied to the leaves of plants, they enter into the cells by some yet undefined mechanism, and they trigger resistance in the plant to any virus that possesses the complementary target, and that this results in degradation we spoke of earlier. Degradation of the virus and suppression of the replication of the related incoming virus. The effect of exogenous double-stranded RNA on virus resistance in various plant species has been analyzed with various viruses and different crops. It has worked. The technique, plants have been found to show delay in development of disease, reduce symptom expression, and a reduction in the number of infected plants, and decrease in virus replication. Kinks in the practical application, these have all been done in greenhouse studies, and the kinks in practical application of the technique to the field are still being sorted out. In recent years, scientists have harnessed the unique properties of viruses not only as tools for protecting against other viral and pathogenic infections, but also as agents for controlling insect pests and combating bacterial infections. Bacola viruses are one such group of viruses. Bacola viruses are known to regulate insect populations in nature. They exhibit a precisely defined, narrow range of hosts, making them ideal candidates for species-specific and targeted insecticidal applications. The strategy behind using these Bacola viruses lies in the ingestion of the protecting virus by insect larvae. And this leads to a fatal infection. As the infected insect succumbs to infection, it releases more viruses, and so infecting additional insects and contributing to the effectiveness of this biological control method. Some 50 registered pesticides based on Bacola viruses are available on the market. One example is Gemstar. Gemstar is labeled as a biological insecticide for the control of corn earworm. Management of this pest with chemical insecticides has become increasingly ineffective due to development of resistance. Gemstar is used extensively against corn earworm infestations in China and in Australia. Viruses hold great promise as therapeutic agents for treating specific bacterial infections. Phages, remember phage is another term for viruses that infect bacteria. So phage therapy has been extensively researched in Russia and Poland since the 1920s to combat bacterial infections. The field is rapidly advancing and is driven by growing resistance to antibiotics. The strategy employs viruses, phages as targeted antibacterial treatments. The viruses employ the lytic cycle. That is, they attach to the bacterial cells, they infect the bacterial cells, and ultimately cause the destruction, the breaking open, the destruction of bacterial cells. Although phage treatment is not approved for use in this part of the world, it is available in Russia and Poland. Intestiphage is one example that is accessible to the public without prescription in both countries. Intestiphage is used to treat bacterial infections affecting the gastrointestinal tract. It consists of a cocktail of phages that target some 20 different pathogenic gastrointestinal bacteria. Similarly, the exploration of oncolytic viruses, which selectively infect and eliminate cancer cells, is underway as targeted and tailored treatment of cancer. Unlike chemotherapy and radiation, oncolytic viruses go after cancer cells while largely sparing healthy cells. These viruses are increasingly considered as a less toxic and more efficient approach to treating cancer. In 2015, the FDA approved the first oncolytic virus therapy, T-VEC, for treating a type of skin cancer. T-VEC is a modified herpes virus that specifically targets cancer cells, inducing their lysis. This lysis of the cancer cell inactivates the patient's immune system, alerting it to the presence of cancer cells, and triggers an immune response against non-infected cancer cells elsewhere in the body. T-VEC is administered through direct injection into visible tumors. To date, 4 oncolytic viruses have been approved for the treatment of cancer globally, but T-VEC remains the only widely approved therapy. Whether directed at harmful bacteria or cancer cells, therapeutic viruses function as microscopic guided missiles that enter and destroy undesirable cells. But in contrast to oncolytic virus therapies, where treatment is based on virus replication and cell death, non-replicating viruses are being used as vectors for corrective gene delivery or gene therapy. The goal of virus-mediated gene therapy is the delivery and expression of therapeutic genes to desired target cells to restore the function of a defective gene. This is typically for the treatment of single gene disorders. Single gene disorders are ideal for gene therapy because they involve a disease-causing variation or mutation in only one gene. Viral gene therapy uses the natural capacity of viruses to protect the encapsulated nucleic acid from degradation and to deliver the functional gene to the DNA of cells. As shown in the slide, there are 2 types of gene therapy: the ex vivo, which involves altering cells in the lab and then returning them to the patient, and the in vivo, the in vivo approach that sends the virus vector with the therapeutic gene, the functional gene, into the target, to the affected tissue. Luxturna is a prescription gene therapy product. Luxturna was approved in the US in 2017 to treat patients with retinal dystrophy. Retinal dystrophy is characterized by the degeneration of the retina. It is caused by a single mutation in a gene, the gene RPE65. Gene therapy for retinal dystrophy involves the use of a modified adenovirus vector to deliver a normally functioning RPE65 gene to the patient's eye. It's a one-time injection into the retina of each eye. Luxturna has and can improve vision in patients with some viable retinal cells. The drug costs $425,000 per eye, totaling $850,000 per patient. Our policies on pricing and payments need to catch up with these types of innovations. And then there are viruses upon which organisms depend for their survival. These viruses confer beneficial properties in a relationship with their host, allowing the host to thrive in specific ecological niche. The first example in the slide is the tail of a grass, a fungus, and a virus. The fungus colonizes the grass. The fungus harbors a virus, and all three participants in this relationship, the fungus, the virus, and the grass, are essential for survival in soils exceeding 60 degrees Celsius, an environment where they would not individually strive. It was demonstrated that the absence of the virus in the fungus results in the lack of thermal tolerance for both the plant and the fungus. Evidently, this virus, which is named Curvularia thermal tolerance virus, that virus activates genes in the fungus that it infects. It activates genes that are associated with the production of osmoprotectants, substances that help organisms to cope with osmotic stress. The production of these osmoprotectants in the fungus facilitates the survival of all three entities under these challenging conditions. Other plants, viruses such as cucumber mosaic virus, tobacco mosaic virus, tobacco rattlevirus, are able to confer drought tolerance to a number of plants, similarly causing the effect of thermal protectants being produced. And under greenhouse conditions, cucumber mosaic virus conferred cold tolerance to beets. All discoveries that could become useful for organisms in marine ecosystems, these are susceptible to viruses. Phytoplankton, your chlorophytes, dinoflagellates, diatoms, these are the primary producers of oxygen, just as plants are on land. Photosynthesis by phytoplankton is responsible for over half the oxygen on the planet. Phytoplankton serve as primary food source for various marine organisms. Phytoplankton virus infections are major determinants in the geochemical cycles in oceans. When viruses infect phytoplankton, they cause cell lysis, releasing organic matter and fixed carbon into the surrounding water. The process contributes to dissolved organic carbon and becomes available to microbial communities. But the lysis of phytoplankton by viruses serves a dual purpose in the marine ecosystem. On one hand, contributes to these essential nutrients to the microbial communities, and on the other hand, the interactions and lysis act as a natural regulatory mechanism, keeping phytoplankton communities in check by controlling their population sizes. Well, thank you. Thank you for joining in these viral tales. Tales of viruses that make us sick, viruses that are harmless stowaways, viruses that play an important part in our ecosystems, viruses that hold promise as therapeutic agents in treating certain diseases. Remarkable entities, so minuscule and not typically classified as living. We have just begun to scratch the surface in the study of their diversity, in the study of their influence. And this study is not confined to the laboratories. The study of viruses has far-reaching implications for public policy, economics, and societal behavior. Understanding the dynamics of viruses and virus infections informs the development of effective policies, influences economic decisions, and shapes how viruses and communities respond to and navigate through health crises, and addresses challenges associated with products developed from the entities themselves. The challenge lies in determining how to manage the so-called bad viruses and harness the benefits of the good viruses and navigate the dynamics between viruses and our broader economic landscape. Thank you.
As expected, ladies and gentlemen, thank you very much, Professor Tennant. That was a very interesting lecture. I personally learned a lot about the dual nature of viruses. We saw and we learned that they have, I suppose, not only gotten a bad reputation over many centuries, but they are exceedingly useful, and they are at least responsible for 8% of our genetic composition. 8, yeah. And I'm thankful, as always, that you, our guests, from wherever in the world that you are today, chose to spend some of your valuable time with us. It is always a pleasure. We look forward to having you again for the next in our lectures in our series. And I look forward to hearing and learning a lot more about what happens here at the Mona campus at the University of the West Indies, and enjoy the remainder of the day. And thanks again to Professor Tennant and our colleagues and the team here that made this lecture possible. See you again soon.